and repeated, just like any other polymers that we studied so far. Okay, here they are again, phosphate group. This would be a nucleotide because it has a nitrogenous base. It doesn't matter what nitrogenous base it has. It has a sugar molecule, it has a phosphate, so it is a nucleotide right here. I did draw it for you like that. Right here, a nucleotide. Pentose sugar. And then they're right in the polypeptide chain. Again, beating the dead horse, pyrimidine, there are 3 of them. Purine, I did not erase them because I would like you to know them, the difference between DNA and RNA. The sugar molecule, of course. There are several differences between DNA and RNA. This is the reason they call it DNA, deoxy-D ribose nucleic acid, ribose RNA nucleic acid, okay? Here is animation. Let's see what the animation has to say. You're almost done.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide.
Don't worry about 5' to 3'.
RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
From here to here is a gene. Okay, do not worry about 5', 3'. That's something we'll tackle later on during semester, but these are DNA, another DNA. Let's see, what is this one?
DNA consists of 2 complementary strands of nucleotides twisted together to form a double helix. In the space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the 2 sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with the thymine and a guanine with the cytosine.
Again, don't want to worry
The
about.
double helix can also be represented by a ball and stick model. The simplicity of ribbon model is often used, with ribbons for the sugar phosphate backbones.
Okay. Let's not worry about that one either. These are, I deleted. Bioinformatics uses computer software and other computational tools to deal with the data resulting from sequencing many genomes, analyzing large set of genes, or even comparing whole genomes of different species is called genomics. And then a similar analysis of large sets of proteins, including their sequences, called proteomics. I did make you one page. It's not here. Let's go. Okay, but let's go over this, and then maybe during the lab I will flash it up. So this is a plant cell. Can you guys see this? Yes, you can see it. A plant cell. Question is, where are? Can you already see this? Where are the building blocks that we talked about at the beginning of the discussions for this chapter? Where are the building blocks in a plant cell? This is a plant cell. Where do you find those building blocks? Carbohydrates make up what? Where am I pointing at? Cell wall. Cell wall is different than cell membrane. So carbohydrates makes up the cell wall. And what is the name of the molecule that is in the cell wall? Cellulose. Underneath of cellulose is what? What?
Membrane.
Cell membrane, not membrane. Cell membrane or plasmamembrane. They call it plasmamembrane. Plasmamembrane and cell membrane. What is it made up of? What building block this is made up of? You have to be specific. You cannot say fact. I will not take it during the exam. What is it? Huh? No. Not monosaccharides. What is it? Anybody? Cell membrane is made up of? Nobody? I did not label it, I guess. The copies is not coming up pretty good. Cell membrane right here. Cell membrane is what? Yellow. And the yellow in here is, of course, lipid. Lipid is phospholipid. Is that right? Phospholipid. What else? Nucleus is made up of what? Inside of nucleus. We just talked about it. Huh? Are you guys listening? No? Did you know you have a test on Wednesday? Did you know this coming Sunday, is the last day to drop without a W in your record? And after that, you sign up for the rest of your life with me. Am I right? They might ask you in the spring of 2016, how come? What happened that you dropped biology 1? Later on during semester, you drop it again. Why have W in your record for Bio 1? They look at it. Why? They say, well, I had a teacher. He was not good. Who are you going to believe? You or?
Nucleic acid.
It's a nitrogenous-based nucleic acid. We just talked about it. It's a nucleic acid, DNA. You guys were sleeping this morning. Right? Or no? You were not sleeping this morning? You were awake? All of you were awake? I saw your eyes were open, except Reno, his eyes was closed. But the rest of your eyes was open. If he doesn't answer it, he has the right. What else? The deposits, lipid deposits. The vacuole here is made up of carbohydrates and proteins. So you have protein deposits here. How about animal cell? Right here. Animal cell. Do they have cell wall?
No.
No. What is this? Huh?
Cell membrane.
Cell, plasma membrane, cell membrane, made up of what molecule?
Lipids.
Phospholipids. 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 And then, of course, it has some carbohydrates in there too. What else? Nucleus. Nucleus is made up of what? Nucleic acids. And then all of the cytoplasm is made up of what?
Carbohydrates.
Cytoplasm? Protein and water, which is a salt gel, colloid. You remember that in your notes? You wrote it down. So, and then you have, of course, you have glycogen deposits and other molecules in there as well. So, that's, let's, is there any question about what you guys need to know for this material?